This is a production of Cornell University.
Stem rust caused by Puccinia graminis for me, especially Irtici, is the most damaging disease of wheat. It's an obligate biotroph and changes a healthy-looking crop to a stem covered with black spore in weeks prior to harvest. And it can also cause yield loss of up to 100% when it's under severe epidemics when a susceptible variety is grown. And this picture is a picture taken in Ethiopia in 2013-14 due to an epidemics which caused 100% yield loss on a popular variety called Digalu due to an emergence of a new race. The fungus is heterocious and it has a complex life cycle with all the 5 spore stages. The main host is wheat and barberry is an alternate host. This is the life cycle of the pathogen. As you see from here, infection starts with spore, which is a sexual spore, then wheat to wheat dispersal is through the basidiospores, which are asexual spores. Towards maturity, the fungus is overwintering as teleospores, which undergoes karyogamy under conducive environment and infects the barberry plant as basidiospore, and then it undergoes its sexual cycle and turns to pycnospores and ascospores and continues the cycle. Sorry. So stem rust is a global problem where wheat is grown in the world. This map is showing the stem rust survey in 2017, and areas with red circles are areas with greater than 40% stem rust severity. So as you see from this map, it's all over the world, and 40% severity is huge enough to cause a damage. The spore of PGT has the ability to disperse long distance. This is another challenge with this pathogen. So as you see from this map, the spores are, wind-blown spores are traveling from their long distance. As you see from this map, they are traveling from the southern parts of US to the northern part and cause the damage. And this map is showing the dispersal of UG99 from Uganda in 1999 to Kenya, Ethiopia, Yemen, and the rest part of the world. So this nature of the pathogen makes it challenging for management. And the common management options are fungicide spraying and genetic resistance. As we all know, fungicide spraying is costly and it's not eco-friendly, and the pathogen may develop fungicide resistance upon continuous use. So genetic resistance is economical and eco-friendly to use. And there are 2 classes of genetic resistance, and it's seedling resistance or origin, which is race-specific and expressed at seedling stage, and adult plant resistance, which is expressed at adult plant stage, and it's more durable than seedling resistance. So we did seedling evaluation against 4 important races, and for this, we have got a panel of 283 genotypes from CMIT, and we evaluated for seedling response in University of Minnesota BCL3 facility against 4 races. The TTKSK is the original UG99 race, TKTTF is the Digalu race, and JRCQC and TTRTF are special Durum races. Seedlings were planted in the university facility there and inoculated with a spore suspension 7 days after planting. Then scoring was done 14 days post-inoculation based on a 0 to 4 scale of Stackman. And this scaling method has plus and minus based on the spore size. And there are some symbols here for hypersensitive response. And we linearized that to 0 to 9 scale according to Zank to make it easy and for analysis. So the distribution of seedling response of the panels against the 4 races, as you see, there are different levels of resistance from race specific to broad spectrum resistance. As you see from these lines or genotypes which have less than or equal to 6 are categorized under resistance, and those above 6 are categorized under susceptible. So we are expecting adult plant resistance from the seedling susceptible. So this is a table showing the level of resistance from 1, 2 to 3 to a combination of 4 races. So there is ranges of seedling resistance in here. So having combined resistance is good enough to combine it with other sources of resistance to get enough level of resistance for stem rust. So we asked the genotyping is done in North Carolina, USDA. We haven't got the data yet. We'll be getting it soon, but we asked them to run the markers for the most known SR genes. They run for the SR2, SR13, LR46. And when I see none of our materials have the SR2 gene. SR2 is a known adult plant resistance gene, and it's durable and it has been effective for more than about 100 years I've seen on the literature. And SR13, the majority of the Durum genotypes we have, about 70% of them have SR13. This SR13 gene is it's more prevalent in Durum and is effective to all the 13 lineages of the UG99. But this gene is defeated by the GRCQC gene. I mean the GRCQC race. And LR46 is an adult leaf rust, an adult plant resistance for leaf rust. And this gene is tightly linked to the adult plant stem rust resistance gene SR58. And this is an indication that we may have some APR genes or some seedling resistance genes. And about 30% of the materials have both. As you see from this graph, we have high level of resistance for the TK-TTF and TTK-SK race. This is because compared to the Durum races, to the J race and the TTR-TF race, these two, we have high level of resistance because, as I mentioned earlier, the Durum genotypes we have in here have more SR13, and these two races are virulent to SR13. So this is also the correlation between the response between the four races, as like this, it's this one, the TTK-SK and the TK-TTF response is high. This is because of the resistance of the Durum to these two races, but these races are still virulent on another more frequent gene, SR9E. That's why we included them in here. And I tried to fit linear mixed model using LMAR function of R, considering the genotype and the replication as a random effect, and estimated heritability for single race from 0.61 to 0.91, which is high enough, and it makes sense because it is race-specific or qualitative resistance. When I fit a combined analysis for the genotype by race interaction to the previous model, the heritability of about 0.585 is estimated. This is also pretty high, and it makes sense because this is qualitative resistance. And we evaluated the panel of the same genotypes for adult plant resistance, including 3 checks in randomized complete block design in 2 reps in Ethiopia and Kenya. And this is how the nursery looks, and their genotypes were planted in 2 rows of 1 meter length and surrounded by spreader rows. The spreader rows are a combination of universally susceptible varieties with equal proportions. There are 4, and their spore suspensions will be inoculated on the spreader rows at 1 meter interval. And scoring is based on severity and response. Severity is based on the modified Cobb scale method, which is estimating the area of stem covered, the percentage of area of the stem covered by pustule. And response is based on Rolfsen. R stands for resistance, MR is moderately resistant, MS is moderately susceptible, and S is for susceptible. So sometimes there is a combination of response, and in this case, we are trying to change the 2 by combining 2 coefficient of infection by multiplying severity by a constant for the response. And this is the data we are using for analysis. So this is the distribution of the stem rust response in Ethiopia in off-season 2018, and it's pretty normal. And this is the main season distribution, which is a bit skewed, and tried to normalize it by square root transformation to fit linear model. And for the off-season, I fitted linear mixed model using ACE-REML and did spatial correction. And in this model, I consider the genotype effect as a random and the rep effect also as a random. And I included the Coleman effect because it's a wild test for Coleman effect is significant, and a heritability of 0.71 is estimated for this one. And for the main season, the spatial correction didn't work well, and I fitted the LMER function of R, and considering the rep and the genotype as a random effect, and heritability of 0.64 was estimated. I received this data by mail, and I have to check this again. And this is a distribution of the response of the panels in Kenya. And in Kenya, the predominant race are the UG99 race, TTK-SK, and the TTK-ST race. So as you see, it's skewed to the right, and there is more resistance to the Kenyan race than the Ethiopian race. This is because of the Durum race have the SR13 gene, which is resistant to the Kenyan races. And I tried to normalize it to fit linear model by square root transformation. And like the previous time, I fitted a linear mixed model using ACE-REML and did spatial correction. In this case, both the Coleman and row effect are, the wild test is significant, so I maintained them in the model and estimated a heritability of about 0.77, which is so good for field resistance. So in summary, stem rust is the most destructive disease of wheat. The race TTK-SK, TK-TTF, GRCQC, and TRTF were selected based on their virulence on commercially deployed SR genes. And ranges of seedling resistance from single race to broad spectrum were observed in the panel. And the field resistance we observed could be all stage resistance, that means it's qualitative, or due to the presence of adult plant resistance. This we'll see later after getting the marker data and did the analysis. And resistance can be boosted by combining this SR13 or LR46 with other SR genes. The next plan is to do GWAS analysis after getting the marker data and scoring off-season nursery in Ethiopia and planting and phenotyping in Ethiopia and Kenya. Okay. At last, I would like to thank my special committee, Dr. Mark Sorrells, Gary Burstum, Dr. Gary Burstum, Dr. Marcellus, and the DGW project of Cornell for funding, Dr. Ronnie Kaufman, Kelly Arthur, and Linda Magrilis, and my institute, EIR, and the DZARC pathology and breeding team, especially Dr. Badada and Ashannafi Gammachu, who are taking care of my nurseries in Ethiopia till I get there. And I would also like to thank Dr. Karim Amar from CIMIT for providing the mapping population, Dr. Bekkele and Dr. Abi, Dr. Bekkele and Dr. Ayala Badi both for processing my import of seeds and the quarantine process there. Dr. Mamdip and Ruth Wainera in Kenya for phenotyping in Kenya. Dr. Pablo Oliveira from University of Minnesota. We did the Seedling evaluation together. And the USDA North Carolina for genotyping, Dr. Gina Jared Kimberley, Dr. Brian Ward. And I would like to thank also my lab mates, Janik and Mark Sorrells in Janik's lab group, Daniel Sweeney, Dr. Chantal, Martis Elitag, and Dr. Albina, Carl Kunz, Travis Roney, James Tanaka, David Bencher, Amy Fox, and Dr. Jean-Luc Janik, Dr. Mahmoud Somo, Moshod Bakare, and Itaraju Jr. Thank you all for listening. And if you have questions, I would like to take some.
This has been a production of Cornell University, on the web at cornell.edu.